ATP is cellular energy. Right? So passive transport does not use ATP, while active transport does. Right? So passive transport does not use ATP, active transport uses ATP. We're going to talk a little bit about different types of transport. We'll talk about diffusion. Okay, we kind of mentioned in lab, we went experimented in lab. We'll talk about carrier mediated transport. It could either be passive, where no ATP is used, but also use carrier mediated methods in active transport as well. Carriers. Vesicular transport is when we transport things in bulk. Okay? We can put it in vesicles. Vesicles are big, large sacks. Like lysosomes, peroxisomes, they were kind of like this. They are vesicle bound cells. Okay? We're not really transporting, they're present in us. Okay? Golgi, what was the function of Golgi? Golgi packaging, right? So they will modify the package and package it into these vesicles in the center. So vesicle transport is using large or transporting large number, could be also large molecule, bulk transport. So what is the definition of diffusion? Movement of particles from high to low temperature. Do we need ATP? Please don't just say they don't require energy. Because what is the energy of movement for? Do these molecules have kinetic energy? Yes, they are moving. Okay. Please specify they do not require ATP or cellular energy. If there's cellular energy, that's it. Don't just say they don't require energy. Because we'll talk about it. What does temperature do to molecules? They change the speed at which they're moving. They actually increase the energy, which is why diffusion also happens. So diffusion is the movement of substances, particles from high to low concentration. The difference in concentration is called concentration gradient. They stop diffusing, they don't stop movement. Okay, they stop diffusion when we say when they reach equilibrium. Equilibrium is a term that's used in multiple places. It just means equal. In this case, equal square. Okay. So here they're dropping this cube of this, we call it a dye. Okay, it's dropped here, it's spreading, diffusing. Okay, and then once it's fully spread equally, that's equilibrium's reached. There is no huge once there is no movement, just like we said in isotonic, there is movement of water, but no net. It's equal. So it's uniform, there is no concentration gradient, so it does not diffuse. Let's also remind ourselves what are some things that can affect diffusion rate. Temperature, right? If we heat it up, what will the diffusion rate do? Be higher or lower or same? That's good. Size of particles. So we compare two particles. Hopefully you recognize, or you read, or looked up. Which one was heavier, potassium permanganate or methylene blue? So which one would we say is heavier, the one that moves further or moves less? The one that moves less will be the heavier one. So we'll have to change the back to your notes to see if it reflected it. If you Google the molecular weight of which you have to get the molecules. One's around 300 something, one's around 100. Lower hundreds, high hundreds, lower 300. So there is a difference in molecular mass, difference in size. So higher, the bigger the molecule, the slower the hopefully we saw that. Which one did we use for the water? Was it methylene blue? That was the heavy one. We used it because we didn't want to spread too fast. We used, potassium permanganate was spread too fast. So we didn't want to do that. Okay, what else? Concentration gradient. So we talked about how when we get to equilibrium, it will slow down. So hopefully we also saw that initially they spread faster, hopefully, right? And then they kind of slow down. They still diffuse, but the diffusion might have stopped. Especially in the refrigerator, you might not see a lot of change from say 45 to 16 minutes, right? Because that was because of temperature, but also the diffusion rate does decrease as concentration gradient. We started with everything in one spot, now it's in a bigger spot, right? So it's moving slower. What was the third thing? The fourth thing?
Density of the medium.
Density of medium. So we looked at agar, we looked at water, right? Which one moved faster? Water. Why did it move faster? Less dense. Right? The agar was a lot more dense. Good. So now let's talk about across the membrane itself, right? I started that conversation, right? We talked about how things can move across this. Depending on the properties, it can just move through the membrane or it needs some sort of proteins. Okay? So we say it's simple diffusion. What are some things that can move freely? Okay, it has to be lipid soluble. Alcohol is actually lipid soluble, which is why it can absorb very quickly. Fatty acids, steroids, even gases like oxygen, polar or non-polar. O2. Non-polar. Let's remind ourselves, what does it mean to be a polar molecule? Why? What does it mean? You were correct the first time, that's the usual format, but it's not necessary that it will be carbon, it has to be oxygen and hydrogen. But what does it mean to be polar? Why is there a polar molecule? Why does it matter?
Because the electrons aren't shared equally.
Because they're not shared equally, so there's a charge difference in the two sides, right? It's not completely charged, but it's partial charged, right? O2 gas, is that going to be charged? Is it going to be polar? Why not?
Because it's the same molecule.
Same atoms, right, in that molecule, so neither will have a strong. Okay, while water is polar. Why? What is water? What are the elements in water? H2O. So why is water polar? Yeah, oxygen actually will pull the electrons closer to it, while hydrogen's got a lonely proton, so it doesn't have a strong pull towards its electron, so the electron spends more time being atoms. What is a hydrogen bond? Is it a bond within one molecule? No, between molecules, intermolecular bonds. So one molecule attracting another molecule. Partially negative side of one molecule will attract the partially positive side of another molecule. That's hydrogen bond. By the way, DNA, that's holding those two strands of DNA together, are hydrogen bonds. That's what's holding those two strands together. So dissolved gases, oxygen, CO2, they're non-polar, they will simply diffuse through the plasma. That's called simple diffusion, when nothing needs to help them out. No proteins are necessary for the movement of these molecules. They have to be small, they have to be lipid soluble. Lipid soluble means they have to be non-polar. Okay? They can just diffuse through vitamins. Lipid soluble vitamins will just simply diffuse through that plasma. The others, we need some help. Okay? We can, if it's small enough, we can use facilitated diffusion. Again, what's the difference between passive transport and active transport. Which one uses ATP? Active transport. Is diffusion passive or active? Passive. By definition, diffusion is passive. Which means facilitate diffusion is passive. All it needs is something to help. That something is a protein molecule that connects the outside to the inside. It could be a channel, which is like, you can think of it like a tunnel through a mountain, right? Or a carrier. I give this example of a ferry. Okay, it's only open on, it's not the perfect example, but hopefully you kind of see, it'll only be docked on one side of the river, not be docked at the same side. So it's only open on one side. That's the difference between carrier and channel. Channels are like a tunnel through, while the carrier is basically only open on one side. Okay? Osmosis. What was the definition of osmosis? Osmosis? Yes, very good for two. If you said movement, you have to say. Otherwise, what word can we use instead?
Diffusion.
Diffusion. So it's a diffusion of water or solvent across the plasma. Can water simply diffuse through? No, you cannot. It's going to use a channel. Okay? There is a specific channel in our membranes that only water passes through. Okay, we call aquaporins. And that's what allows water. Osmosis is basically diffusion of water through phase. Well, filtration, I mentioned here, I know your slide says it as it's a type of passive diffusion. Kind of true, but not exactly. But what is true, it's a passive transport, right? If we were to filter a mixture of solution through a filter paper, there is no ATP there required. So it's still passive transport. We can kind of think of it as passive diffusion, but please note, it's not a membrane here, it's actually kind of
Bless
a
you.
wall, a capillary wall itself. It's not the cells itself. Okay? That's why it's different. Okay? But it is passive. There's no ATP at all. Okay? So let's talk about some channels. Okay. So first of all, channels are basically these tunnels through the membrane. They could be gated. That means it's like, they took off the lever when we crossed the bridge now. It's a electronic toll. But if you went to a parking lot where you have to pay for parking, and then there's a guard there, right? It would have this lever which will stop you from passing whichever way, usually out, until you pay them. Okay? So that is a gated channel. So some condition has to be met for that gate to open on purpose. Okay? While leak channels, they're always open. Okay? Aquaporin is a leak channel. So it's always open as long as there's a concentration gradient. So even without it, water still moves through, but net gain or loss to something like this, a concentration of water. Okay? The gated channels only open or close when something signals. So we'll talk a little bit about them in our nervous system, chemical or electrical signal can open or shut these gates. While leak channels are always open. Okay? So things will move in and out as long as there's a need for concentration gradient, but we're very quickly from the lots of people move in that direction. Any questions? Channels basically connect, if I was to draw it out, basically a tunnel through the plasma membrane. Yeah, I'm trying to draw a three-dimensional structure, two-dimensionally, and if you think of it as a tube. So this way, this top for substances to avoid these liquids. The water, for example, uses a channel protein, a leak channel. So let's talk about osmosis. We mentioned diffusion of water through a semi-permeable membrane. Please remember it's a concentration gradient of water. Whenever we talk about solutions, how do we describe solutions? As a percentage of solute, right? Solvent is the other part of it. So keep reminding yourself, if the solute numbers are higher, that means the solvent concentration is lower. And the opposite of that. So basically, water moves to an area which has a higher solute concentration. Why does it move to an area of higher solute concentration? Because it has a lower solvent. It's moving down its concentration gradient. It's moving down its concentration gradient. So solvent, water is a universal solvent. Okay, then we went through this in lab. Some people only will talk about today, others hopefully at some time to think about. So let's remind ourselves, isotonic, hypotonic, hypertonic. What does iso mean?
Same.
Hypo,
Low.
less, and hyper.
High.
In terms of tonicity here, what is the main import or main explanation of it?
The concentration of the solute.
Solute content. Solute content. So there's a difference in solute concentrations. We would call them either hypo or hyper. If there's no difference, that's iso. So same solute, which means same amount of water, so no net movement. Please don't say no movement of water. The water doesn't move, no getting or loss. Getting or loss. Well, if we place a solution in a or cell in a hypotonic solution, which direction will water move? If we placed our cells in a hypotonic solution into Y, what does hypo mean again? Hypotonic mean again? That's solute, which means what about solvent? Higher solvent, right? Higher water content outside, so the water will go into this. Opposite is true. We have hypertonic solution. We have more water inside now, there's less water outside. More solute, less water. Here will be images. This is an electron microscope image. You can see the nice donut shape of our hydrogen cell. But if we put it in a hypotonic solution, less solute, more water, water will come into the cell. Okay, and it keeps getting bigger and bigger depending on how hypotonic is. For example, pure water, it will just explode out because you will never have equilibrium with the outside. Always be hypotonic on the outside, so it just keeps coming. So this is just a while hypertonic means more solute, which means less solvent. So it's going to lose water, it's going to shrink. That's crenation. Okay, that is a crinkled cell. Cells, but they still got nuclear contents, but the water is being taken up and swooshes inside. Not nuclear contents, cellular contents. Hypertonic means more solute, less solvent, so water. So which one of these two is seawater to us? Hypertonic. Dehydrate ourselves. More solute, less solvent. So here's the image showing 3% in HCl. Roughly, what's our concentration in our cell? That's the solute concentration of our cells. It's equivalent to 0.9% DCO, right? Which is what we use in our normal saline because it's isotonic to our regular cells. When we dehydrate it, we still have normal saline, say the water will still go into our cells. When we're dehydrated, is the solute content going to be high or low?
Low.
It's going to be high.
High.
So the same amount of solute, less water.
Yeah, we have less water, the percentage solute is going to be high. This 0.8 can be like 1.1. Okay, still with more water, more less. Equilibrium. Or if we put blood cells into a hypotonic solution, they will fill up. If it's very hypotonic, they will move reverse. Red blood cells goes to hemolysis. These pink cells are called innate cells of formation. Any questions? So continuing on, active transport. So this requires ATP. Okay? Not dependent on concentration gradient. Sometimes there's a concentration gradient, but we still need ATP. For example, large sized molecule. Okay? It cannot move through a simply or through these membrane proteins. There too, you may have to use ATP. Does not depend on that. So it would be down the concentration gradient, but it would be against concentration gradient. And the one that we're going to look at is the movement of sodium and potassium. I don't know if I mentioned already, where do we have more sodium, inside our cells or outside our cells? You may even know if you look at your blood results. Yeah.
Outside of our cells?
Outside, if you look at your blood results next time, look at the normal ranges, that's going to be in the hundreds, roughly about 35 and 45. The sodium or potassium numbers. Oh, by the way, where is that numbers from? When they do biochemistry on our blood. What part of our blood are they running? It's a plasma, right? So it's extracellular fluid. Okay? Potassium is around the 4 millimolar mark. Okay? It's about 4.5. It goes from 3.8 to about 4.5. But on the inside, we have a lot more potassium. It's kind of reversed. Sodium's about 4, potassium's about 20. Okay? So inside, we have more potassium. Outside the cell, we have more sodium. And that's the way we like it. We want it to equilibrate. Okay? So we will have what's called the sodium-potassium pump. The first thing about a pump is active transport, ATP. Okay. Next thing, it's moving against concentration gradient. So which direction would this sodium-potassium pump move sodium? Into the cell or out of the cell?
Into the cell?
Where do we have more sodium? Outside.
Outside.
I said it's going to move against concentration gradient. So it moves more of them out of the cell. Okay? That's what we need those ATP for. It also moves potassium against its concentration gradient. Which direction would it move potassium then? Into the cell. Inside has more potassium. We want to maintain that, so it keeps moving potassium into the cell. Okay? Why does it have to happen? Because we also have sodium and potassium leak channels. What does leak channels mean? Always open. Active or passive transport?
Passive.
Passive, right? Leak channels always open, or passive transport. Which direction would sodium want to move? Into the cell. So you have these leak channels. These are present in our cell. Okay? Sodium will move in the cell. You also have potassium leak channels. Potassium will move out. It will equally have equal amounts on both sides. You just leave that free. Okay, we do not like it. We have a sodium potassium pump that helps to maintain. Please note it's going to pump. There's also another thing, detail. It's also not equal number. Okay, the first thing, it's going to move sodium out of the cell against its concentration gradient. It's going to move potassium into the cell. Okay? In our nervous system lab, we'll talk about how the inside of our cell is slightly negative than the outside. This is one of the reasons. Okay? What's the charge of a sodium ion? Again, if you don't remember, look at your periodic table and try and predict that. It's +1. Why is it +1? There's 1 electron. How do we know that? Right. What's potassium's symbol? Okay, what will it do? Also lose what charge will it have? +1. So we are losing 3 positive charges, but we're only gaining 2 positive charges. So every time the sodium and potassium work, we're losing a positive charge, which means the inside becomes more negative. Okay, we'll come back to this in a minute. This is another important reason why we are rejecting it. Not the only reason. Sodium potassium pump moves 3 sodiums out against its concentration gradient, moves 2 potassiums in against its concentration gradient. Want more sodium outside, want more potassium inside. Okay, very key. Please underline however you want. Just make sure I'm just going to talk about this a lot, not just in our end course, even in parallel course, even the 7th class. No, because you're losing more each time it goes, and that's another reason why it's more negative. A charge is a movement. Yeah, in the cell.
Oh, okay.
Yeah, because we're losing 3 positive charges, we're getting 2 only back. So basically, every time it works, we're losing a positive charge, so inside is left to be more negative. So vesicular transport, first of all, vesicle is basically a membrane balance. It can move large amounts of stuff. It is an active transport process. It requires ATP. Okay? Depending on which direction it goes, we can say it's endocytosis. What does endo mean? Into. So this is bringing stuff into the cell. And then opposite will be exocytosis, when we're pushing stuff out of the cell. Okay? Exocytosis is opposite, to move material out of the cell. We do that in large amounts of material or large sized particles is what it's vesicular transport. Here what happens is the membrane here is basically this. Okay? We will fuse those, that membrane will fuse with our plasma membrane, only the contents get switched. Same thing with endocytosis too. It's the same membrane part that becomes part of this vesicle. It's just basically reusing material. Okay? Here we will not lose the membrane, we just will lose what's inside of these vesicles. For example, mucus is produced this way, no discharge of exocytosis. Almond mucus, waste products are discharged by exocytosis. Endocytosis into the cell, exocytosis out of the cell. One thing requires heat. Let's look at a little bit of types of endocytosis. We can have what's called receptor mediated. That means there's a receptor, something has to trigger for this to be swallowed. Okay? We kind of use that to swallow in our immune system to swallow up bacteria, for example. Ion transport, interesting enough, iron is just an iron, there's also an ion. That's so small, but we still use membrane receptor mediated endocytosis. Okay, so trying to set that much chance for iron. Will iron move freely through? If you do not know, iron's charge is Fe2 plus or 3 plus 2 ion. Why will it not move freely through our plasma? Ions, I didn't ask that question. I just asked. Why do ions not freely move through our membrane? Any idea? Yeah, because they're kind of polar, they're charged, right? So charged molecules will not freely move. Pinocytosis, pino is in reference to liquid fluid. It's like this cell is drinking. Basically, it's swallowing up all the extracellular fluid, contents and everything together, just taking it into the cell. Right? Phagocytosis is cell eating. That's actually swallowing up pieces of cell or cells themselves, large molecules. Okay? Generally, when we do that, we're going to fuse it with lysosomes. What's the function of lysosomes? Digest. So they can actually digest whatever we do. So for example, bacteria, if this is our immune cell like a macrophage, for example, swallows it up. Lysosomes will bind with this. It will break the contents of that. Okay, some of it will be absorbed. And we can use DNA, RNA, proteins, but as long as we break them to monomers, we can use them. We will use them. Okay? And some substances we cannot, that will be discharged. Any questions? So membrane transport, active versus passive, that's the first thing. What is the difference? Active is
Using
uses ATP, passive does not use it. Diffusion, whether it's simple or facilitated, another subset of a facilitated diffusion. Osmosis, filtration also is passive, does not use it. When you do require ATP, that generally you're moving against the concentration gradient or moving dark substances like a very important example of active transport is swimming for our sodium. Which direction does it pump sodium? Out of the cell, potassium into the cell. How many sodium? 3 sodiums out. How many potassiums? 2 potassiums. So cytoplasm we can separate to organelles and then cytosol. Organelles we look at a few in the lab, we'll look at them again, talk about them. Right? So cytosol is our intracellular fluid. It's got water. It has things dissolved in the water. Okay? Yeah, stored nutrients I mentioned about glycogen can also be found outside the body as we kind of dissolve or as a granules. While organelles, you can think of them as similar to our organs, right? How would you describe an organ? Yeah, a bunch of tissues coming together, 2 or more tissues coming together.
Forms a certain function.
That form a certain. Here, 2 organelles are like the organs of the cell. Right? They are also structurally different and functionally very specific. Okay? So we can divide these organelles to either non-membranous or membranous. Basically, do they have a membrane or not? Non-membranous do not have membranes, membranous have membranes. When we think about a membranous, can we state a few membranous structures, if you will? organelles. Mitochondria does. The rough ER, smooth ER, yeah. Nucleus. It is, yes, it's still an organelle, but we give it as a nice special organelle. When we talk about organelle, in the conjugal site, in cell, we separate out the nucleus and we say, oh, yes, it would be an organelle too, because it does have membrane. It does. Okay, so just before we talk about the membranous ones, let's look at some non-membranous ones. The first one we're going to look at is a group. It's called cytoskeleton, right? Just like you and I have a skeleton that helps to keep our body, give its form, help it move. Cytoskeleton is these structures within the cell that have a similar function, right? Have structure, have the capability of helping them to move. Okay? We have really three types of cytoskeleton. We have microfilaments, intermediate filaments, and then microtubules. I don't know why this is listed separately as part of our microfilament. I think your book has it as four. It's really three microfilaments. The thick filaments are microfilaments. The thin filaments we'll talk about. But these are the three types of cytoskeleton. So microfilaments are the thinnest out of those three. Okay, so going down, this is the thinnest, that's the thinnest. Okay. So microfilaments are the thinnest, and they help to move substances. For example, actin. I think, yeah, this is why they separate, but most books do not separate this way, by the way. It's still effective for us to talk about it. Microfilaments, these actin and thick filaments, the myosins listed here. Okay. These help for muscle cells to be able to move because of the interaction of the thick and thin filaments. We'll talk about later. What else do we talk about? They also help to attach different things to a membrane. In terms of intermediate, the only one that's useful is keratin filaments. That's helpful in strengthening cells. It actually helps bond cells, neighborhood cells. It's like the trusses in buildings. Within the cell, but the buildings may not be the best, but if you're thinking about buildings, the walls attaching to the ceiling, for example, right? They do help to keep the whole cell together and attached to the walls. While microtubules, what do you think tubules might be? Hollow, tube shape, yeah. Okay. They're mainly made of these proteins called tubulin. Looks almost like beads around this tube. Okay. It does help strengthen rigidity. This is one important one. It helps to move organelles. Okay. We'll talk about in cell division. What structure helps to move chromosomes in cell division? We gave it as the centriole. We'll talk about centriole and centrosome. Centrioles are the larger one. They'll actually produce the microtubules. The microtubules that attach us them and then they're moving these. That's the spindle of cell division. So centrioles, centrosome. Previously they were, we call the centrosome centrioles the organelles. But if you remember from the model, what was the centrosomes? What did I say was centrosomes? They help deciding what they look like in the model. There are those black with two, they look like little cylinders. Okay, those two cylinders are actually the centrioles. The whole thing together, we call them centrosomes. But they have common function. Okay, they will help. They'll produce the spindle fibers. These, I know they call them different names. They're still microtubules. Okay, they produce microtubules. You can think of them like the fishermen trying to catch fish. The fishing line is the microtubules, the spindle fibers that extend out from the centrosome to grabs on the cornerstone. We also have structures called cilia. Cilia are little microtubules. They're hair-like, they stick out of the plasma membrane. So if you have, again, I'm drawing a two-dimensional cell, and the nucleus, cilia are little. In humans, cilia just move substances across the cell membrane. Okay? We have cilia in our respiratory tract, we have cilia in our neurogenital tract, or rather genital tract. Right? In cilia in our respiratory tract basically will move mucus. Okay? Mucus is what traps all the dust that comes out. We're producing mucus all the time. We're producing mucus all the time, it traps dust particles. These cilia will slowly move it up towards our throat. Most of the time it just gets swallowed, we don't notice it until we're producing a lot of mucus, then we may notice it, or when the cilia get full, right? So why is smoker's cough present? It's because it's more black mucus, right? If you've ever taken your hand with some hair on it, close to a very hot object, what happens to the hair? Sings, it kind of burns. That's what's happening when smokers smoke the hot air in, the cilia gets burned, so they're not moving the mucus little by little up where they think the mucus is going. So run off the lungs, they cough, do it like that. That's what the smokers are. Good things is cilia do come back when people stop smoking, but we do have other damage which may not be reversed. So cilia basically move substances across the membrane. Humans, we do not move cells with cilia. Bacteria do use cilia for cells. Okay? We just move fluid or substances across the surface. We also have a long microtubule called flagella. Oh, this is an image of cilia. Okay, that's our cell. And the cilia just have some moves. Flagella or flagellum is a microtubule, long microtubule. The only human cell that has it is the sperm cell that helps to propel the whole cell. Okay? Some microorganisms have flagella and cilia, and they can use it to move. But here, it actually will move the whole cell. Okay? While cilia move substances across the cell, so it's going to move the cells themselves. Well, here I'm putting it here, but please note, these are not microtubules by themselves. Okay? If I draw side by side, this will be cilia, while microvilli are foldings of the plasma membrane. That's why I'm drawing with the same mark. Okay? So these microvilli will increase surface area. Instead of being flat, like this, they've got extra surface area. Within the microvilli, however, we do have microtubules. That makes these microvilli a little bit more firmer so they don't totally collapse because there's a lot of cytoplasm and then plasma membrane membrane across the surface. Okay? So microvilli are not themselves microtubules. Okay? They are foldings of the plasma membrane, cell membrane. They increase surface area. Within them, here is orange and brown lines. Those are microtubules, or microfilaments. They're actin filaments that are found within the microvilli. It makes it a little bit more stiffer, that way they can stand a little bit more. That way they're not just collapsing. Right? So you can surface area. For example, our intestines have them, so we can absorb most nutrients that we get from food. ribosomes, we talked about in lab, they're basically going to be producing proteins. But please realize we do have two types of ribosomes, basically whether they're attached or freely floating, right? Where do they attach? To the rough ER, right? They're attached to rough ER, that's the endoplasmic reticulum ER, and the fixed spores. Both of them have the same function. Okay. The next one talks about endoplasmic reticulum. We talked about this in lab, so please read through and go through that. I'm going to highlight rough ER. It produces proteins, but also produces phospholipids. Smooth ER detoxifies drugs. It has all these cholesterol, steroid production, triglyceride production. Okay, we talk about detoxification also. Questions? Finish the rest.